Hello, I'm Saul Ibarra, and along with Wilfredo Garcia and Juan Gonzalez, we will be evaluating the original article, which was titled Direct Numerical Simulation of Cellular Scale Blood Flow in 3D Microvascular Networks. This is our evaluation presentation outline. The problem background evaluation will be covered by me. The improvements on methods and models will be covered by Will. And the analysis of the discussion and conclusion will be covered by Juan. First, I will start off with the problem background evaluation. Future research studies can use what was learned from the original article on complex microvascular networks and expand to cover other blood flow characteristics. This includes cell deformation, lingering dynamics, shear rates, coagulation, platelet activation, which may lead to blood clots, Philipot formations, all which would be helpful in studying blood flow phenomena. Also, the original article analyzes general microvascular networks, where new studies can combine a variety of factors such as blood flow in different parts of the body, like the brain, the heart, or a particular muscle or organ. Future studies can also do a better job in combining in vitro, in vivo, and in silico research with simulation analysis. Researchers can also take a close look at complex microvascular blood flow with emphasis on transportation of medication through the bloodstream, for example, in cancer or heart disease research.
Hi everyone, my name is Wilfredo Garcia and I will be going over the improvements on the methods. The first improvement we would like to discuss will be to utilize the effective and memory viscosity of blood instead of using the viscosity of plasma and hemoglobin. The study uses the latter two in order to build a model and to be able to study the blood behavior within the model. The memory viscosity of red blood cells could have been obtained by using the model developed by Prado et al. It does require some input and output parameters, however, as you can tell from table 1 on the right hand side, they reliably give you the membrane viscosity and the effective viscosity of blood cells. While the researchers do state that using this viscosity will not have an impact on the overall results of the study, we do believe that it will give us a more accurate depiction of blood cell flow within the microvascular networks. The second improvement will be to test for red blood cells with different deformability. This would help enhance the model to be able to determine flow behavior of blood in people with disorders. It also would have medical implications for people that have these disorders such as sickle cell anemia and how these disorders affect blood flow. The third improvement will be to incorporate the wall shear stress induced by red blood cells on the microvascular networks. The current model uses a rigid boundary condition, which means that the walls are not able to expand and retract with different pressures. The wall shear stress plays an important role in blood regulation, and as found by Backlog, shear stress is highly affected by pressure rises, especially in venules. At bifurcations, the wall shear stress gradient is also much greater, so having these important parameters would have been helpful.
Hi, my name is Juan. I'll be going over the analysis of the discussion and conclusion for our article. Some positive aspects were that it reiterated the major aspects of the study, and it clearly explained the effects of lingering at bifurcations of small microvascular networks, such as the reduction of hematocrit upstream and downstream of the bifurcation, the negative pressure flow correlation in the vessels, and the temporal heterogeneity of blood flow in the vessels. Some improvements that we recommend for the study was shortening the discussion to be clear and more to the point. It managed to encompass all the parts of the study, although it could have been shortened to highlight the most important parts and also to clarify the findings to the reader. Another improvement that could have been made to the discussion and conclusion was explain the necessity of studying the blood flow in microvascular networks of healthy cells, which would give more merit to the findings of the study. Another improvement it could have made was to correlate the findings of the study with real-life effects, such as answering the questions of how could the outcome of the study affect the health industry, and could this model be reappropriated to better reflect the effects on diseased cells. And finally, since the networks used in this model are not found in real life, can this model also be used to simulate the actual microvascular networks found in a human body.